Good morning, everyone, and welcome to our first laboratory session. For this morning, we will be doing basic procedures that are being done in the microlab, including gram staining, types of staining, sterilization, types of culture media, to include isolation and streaking procedures. The microlaboratory is a very dynamic yet very challenging area of the laboratory. This is where most of your clinical specimen undergo processing to be able to identify microorganisms found in clinical specimen. You may see inside the laboratory a lot of equipments like your oven, your biosafety cabinets, your loop sterilizers, your staining materials. So you see, it's a very busy and dynamic area. So first of all, we would like to introduce some basic techniques that we employ to be able to isolate microorganisms from clinical specimen. Identification of morphology of isolated organism is basic in a successful workup of microbiologic specimens. However, before we start with staining, to be able to appreciate morphology, a good smear is a basic foundation for every microscopist or microbiologist. Of course, before we can do a good smear, we have to be able to choose well-isolated colonies in solid media or pure cultures in liquid media. Hence, we need to do streaking technique. So what is streaking technique? It is a technique that is used to isolate pure strain from a single species of microorganism in clinical specimen. And then further on, if we have isolated pure colonies, then we can subject them to staining or further workup for microbiologic purposes. Okay, there are some things to remember in doing streaking. We have to use only a small amount of inoculum. When you are sterilizing the inoculating loop, make sure that the entire loop turns orange. So this is what you do when you flame the loop in a Bunsen burner or use loop sterilizer nowadays. So you don't need to use flame in the microlab anymore. So streak the agar media lightly. Flame the loop after each quadrant that you make. Make sure that the surface of the plate is free from droplets of moisture. And then after streaking, the plates are then incubated. The parts of the sterile pipette that comes in contact into the culture must not touch any unsterile surface outside of the plate. Okay, some tips also in doing the isolation technique is to observe aseptic technique. Work near a Bunsen burner or within a biological safety cabinet. A Bunsen burner creates an upward flow of air, lowering the dust and other contaminants from entering the sterile media. Using sterile loops here or nichrome loops, make sure that they are sterilized by flaming them in loop sterilizers or a Bunsen burner to ensure that all microorganisms are destroyed. You do this before and after inoculation of microorganism in solid or liquid media. So here are samples of streaking. The continuous streaking allows a unidirectional flow of streaking across the agar surface, decreasing the population of organism from the point of first inoculum until the last. You also use this technique if we are to save some organisms for further testing or subculturing. The other more common streaking technique to isolate organisms is the four-quadrant technique. This allows to decrease the microbial population for each quadrant until we get pure colonies in the latter portion. This picture will tell you that you have isolated some pure colonies using the continuous streaking and the four quadrant technique. So you will see that in the continuous streaking, you have isolated colonies on the bottom portion, and on the four quadrant technique, you will see pinpoint colonies on the fourth quadrant of streaking. Take note, in four quadrant technique of streaking, you flame the loop successively after each quadrant is done. Now that we have isolated bacterial or microbial colonies, we now begin to prepare a good bacterial smear. So how do we come up with a good bacterial smear? First of all, you choose an isolated colony in the solid media. And gently, if you are taking it from a particular colony, be gentle with the agar media. Just get a loopful in an oil and dirt-free glass slide. Spread the colonies. If it's dry, you can add sterile water and carefully make the smears not too thin and not too thick. After making the initial smear, let it air dry, and then after air drying, you can do heat fixing by using a hot plate or flame it slightly intermittently over a Bunsen burner. This is what you call heat fixation. The smear is now ready for staining. The one on your right is a good smear. There is examples of smears that are made too thick or too thin, and they may not be as optimum for staining and in visualizing microbial populations and morphology. Why? Because in too thick smears, you cannot appreciate singly the detail or the morphology of organism. In too thin, sometimes in viscid specimens like sputum, you may not be able to appreciate the organism at all. Okay, after a good smear, we are now ready for staining. Types of dyes and stains that are used in the microlab includes your basic and acidic dyes. The bacterial cell wall is slightly negatively charged physiologically. So basic dyes in general are positively charged and acidic dyes are negatively charged. Hence, basic dyes are used more commonly such as your crystal violet, your methylene blue, your malachite green, and safranin. They are most commonly used in staining bacterial colonies in smears. We have a lot of staining techniques, and they're different. They are your simple staining and differential staining of organism. In simple staining, you use only one basic dye. You can choose from crystal violet, safranin, methylene blue. For differential stains, it uses more than one type of dye. These procedures react differently with different kinds of bacteria, hence, it will help us distinguish between different kinds of bacteria and their morphology. The most common differential stain that we use in the laboratory is the gram stain. This will differentiate gram-positive from gram-negative organism. Other differential stains include your AFB stains, which stains the organism with fuchsia, pink, or purple, and the endospore stains seen in Bacillus species. Other special stains include your negative staining and flagellar staining. So how do we do simple staining? Initially, we do prepare a good bacterial smear. Afterwards, you may flood the smear with any of the following stains such as methylene blue, crystal violet, or carbol fuchsin. After applying these stains or dyes, The stain is placed on a rack and then washed with water in to be able to rinse excess stain or dye, and then observe under the microscope. This is to show you the simple process of doing simple stain. This is a result of doing simple staining in a bacilliform organism. Note that the morphology of the organism is preserved, and it took the the only stain that we use, which is, yes, you guessed it right, this is crystal violet. Let us now go to differential staining. The most common in the microlab is gram staining. It's a common technique used to differentiate again two large groups of bacteria based on their difference in cell wall constituent. The gram stain distinguishes from gram positive and gram negative groups based on acquisition of the primary stain, which is crystal violet, and the counter stain, which is safranin. In doing gram stain, remember bias. Okay? Apply a primary stain, which is the crystal violet, to a heat fixed mirror. And then you add iodine, which is a mordant. It is usually a metallic compound which binds crystal violet and traps it in the cell. It adds affinity of the stain to the bacterial cell wall. Okay, so after that, we do rapid decolorization with ethanol or acetone. This is an important step in gram staining, as over decolorization may yield a falsely gram negative organism, or under decolorization, which will also yield a falsely gram positive organism. So gentle and rapid decolorization is essential. Then afterwards, we counterstain it with safranin. These are the materials for your gram stain. So label, prepare all the staining materials, prepare a clean oil-free microscope slide. You have to have a heat source for heat fixing, and culture and specimen. You've learned from your lecture that there is a difference between the gram positive and gram negative cell wall. The gram positive, having the thicker peptidoglycan, retains the primary stain, which is crystal violet, while the gram negative organism has a thinner peptidoglycan layer and retains the counter stain, which is safranin. At the point of decolorization, the gram positive yields the primary stain, and the gram negative organism yields does not retain the primary stain, and it becomes colorless. You can see from this slide that there is contrast between the gram positive organism or bacilli, which is color violet, and the gram negative bacilli, which is colored red or retains the counter stain, which is safranin. I added a good link for you to watch and review your gram staining procedure. Let's go to another differential stain or special stain, which is the acid-fast bacilli stain. Acid-fast stain or Ziehl-Neelsen or Kinyon stain acid-fast staining procedure are done to be able to stain or identify acid-fast bacilli. Acid-fast bacilli requires a special technique as their cell wall is composed of mycolic acid. The difference, and you may remember this in Ziehl-Neelsen, remember? Ziehl-Neelsen, Zisling. It means that you apply heat in the staining technique to be able to add affinity of the primary stain, which is carbofuscin, to the acid-fast bacilli. While in the Kinyon method, Kinyon, cold, This does not apply heat upon addition of the primary carbofuscin. However, prolongs the contact time of the stain with the bacterial smear. As you will note in the procedure for Ziehl-Nielsen and Kenyon stain for acid-fast bacilli, there is a counter stain which is methylene blue. The affinity of the acid-fast bacilli to carbofuscin is retained because of the mycolic acid in the cell wall of acid-fast bacilli. The background is usually stained with a counter stain which is methylene blue. Another type of special stain is the negative or indirect staining. What is good about negative or indirect staining is that it only uses one stain. The technique does not use heat fixation of the sample, hence preserving the morphology and does not distort the architecture of the organism. It also employs an acidic stain because the repulsion between the negative charges of the stain and the bacterial surface will not penetrate the cell wall. That's why you call it negative staining. In negative staining, the results yield a clear cell with a dark background, as you can see in the picture on the right. We also have a special negative staining that are used for encapsulated microorganism. And to be able to demonstrate this, the presence of capsule in some microorganism, the microbiologist can mix a solution containing a suspension of India ink and nigrosin. They are both negative stains to produce contrasting background. And then the stain of the bacteria, you stain the bacteria using safranin. And then the chemical composition of the capsule do not accept most biological dyes. Hence, they appear as clear halos surrounding each stained bacterial cell. Another staining technique is the endospore staining. Endospore is a special resistant and dormant structure of organism specially seen in Bacillus species. It cannot be stained by ordinary methods because it is tough, okay, such as simple or gram staining. So usually we use a special technique, and one of the common stains that we use is a Schaefer-Fulton endospore stain. So how do we do endospore staining or the Schaefer-Fulton endospore stain? Usually you use malachite green, which is then applied with heat for 5 minutes. Malachite green will serve as the primary stain and will be able to color the endospores. You wash it with water and use safranin as counter stain. The end result is that the endospore will stain green with red or pink viable cells of the bacilli species. So you can see in this picture, you have the endospore there, and the viable bacilli is red or pink. So we're done with your common staining techniques. Let us now go to a more important procedure done in the microlab, which is sterilization. So as we all know, sterilization is a process by which all forms of microbial life, including spores, are killed. Spores are the toughest one to eradicate when you are trying to sterilize materials that you have to use for microbiology. This is accomplished by physical and chemical means. Disinfection, on the other hand, is the process whereby pathogenic organisms, not necessarily including spores, are destroyed. Disinfection may also be accomplished through physical and chemical methods. So again, disinfection is the process of elimination of organisms, however, does not eradicate all vegetative spores. In sterilization, it refers to a process that eliminates and removes all forms of microbial life present in a specified um, product or region. So we mentioned earlier physical methods of sterilization. And how do we accomplish this one? There are five common physical methods of sterilization. The most common one that we do in microbial loops and needles is incineration, or by burning of the microbial form. Okay. Another is by using moist heat, and this is what we commonly call your autoclaving. And then the next one, you use dry heat, and this is accomplished by a high source of dry heat, which is the oven, and usually entails long contact time for things that you need to sterilize. The fourth one is by filtration method, which is commonly employed in doing filtration or using high particulate air filters. Okay, that you use in your biological safety cabinets, in your mask actually, and last but not the least is by the use of ionizing radiation. Let's discuss them one by one. So, incineration, most common form or method of treating infectious waste. So, usually, hazardous materials are burned into ashes. However, because of environmental safety, we do not usually do this anymore. Usually, things are burned for a temperature of up to 980 degrees. And the limitation is again, it's toxic particles released into the atmosphere. So this is not a usual mode that we require for large quantities of things to be sterilized. Moist heat is autoclaving. It is steam under pressure. This is the most commonly employed sterilization technique in the laboratory and used to sterilize biohazardous trash before we throw them and heat stable objects. So you use the autoclave. Moist heat in the form of saturated steam under an atmosphere of pressure is usually applied. This This is how you usually apply steam pressure in autoclaving a particular culture media to eliminate organism. Uh, the the s- the steam displaces the air in the particles that you are trying to autoclave. So, as we all know, autoclaving is the fastest, simplest physical method of sterilization. So, usually, the gravity displacement type of steam sterilizer is used. So, steam, lighter than air, displaces air inside the sterilizing chamber. So, you usually employ two common sterilizing temperature. The more common one is the 121 degrees Celsius that is used for media, and you apply this for 15 minutes. And then the other one is for 132 degrees Celsius, and you usually do this for 30 to 60 minutes, and you use it for infectious medical wastes. Okay. Dry heat or oven requires longer contact exposure. And then you usually use higher temperature than moist heat. Usually you employ 160 to 180 degrees Celsius. Dry heat ovens are usually used to sterilize glassware, but of course, your glasswares are supposed to be heat resistant. So you usually do this in your laboratory glasswares because they are heat resistant. Okay, let's go to filtration. Filtration is a method of choice for antibiotic solution, toxic chemicals, radioisotopes, your vaccines, and some carbohydrates. All of them are heat sensitive. Filtration of liquids is accomplished by pooling the solution by the use of a cellulose acetate or a cellulose membrane. When it comes to air filtration, and you usually see this in biologic safety cabinet, filtration of air is accomplished using a high efficiency particulate air filters or the HEPA filters. Now we use the ULPA filters or ultrafiltration particulate air filters in your biological safety cabinet. They are designed to remove organisms larger than zero point 0.3 micrometers from isolation rooms, operating rooms, and biological safety cabinets. So by filtration, you do not allow passage of larger particles than zero point 0.3 micrometer through the filtrate or filter. Another physical means of sterilization is by the use of ionizing radiation, by the use of gamma radiation. Usually, it's a cobalt. Okay, the gamma radiation that is used is cobalt. It has a greater energy than your UV light. Hence, it's more effective, and you usually use the uh, ionizing radiation in industrial facilities. Example, you're trying to sterilize disposable plastic syringe, gloves, specimens, and petri dishes. We're done with physical means of sterilization. We now go to chemical sterilization. So, when we say chemical, syempre we use chemicals to do sterilization for us. Okay? So, this is useful for heat sensitive materials such as your plastics, instruments, your endoscopes, and then you can also use ethylene oxide. This is a more common chemical sterilant, and you usually use this in gaseous form. Ethylene oxide alkylates DNA. Hence, since it alkylates DNA, it destroys microbial forms. Okay, so it requires high humidity, and because of this, you have to expose the materials to be sterilized for 4 to 6 hours. Care in using ethylene oxide is that it may cause explosion if it's pure. So usually you mix it with an inert gas, usually with neon or freon. Other chemical means of sterilization, you can use formaldehyde, either vapor or vapor phase hydrogen peroxide. They are both oxidizing agents. They can be used to sterilize your HEPA filters, biologic safety cabinet, and formaldehydes are activated glutaraldehyde. So it's poricidal, it kills spores. So you can use it to sterilize your medical equipment, your bronchoscope, and it also does not corrode your lenses, your metal, and rubber. Another effective chemical sterilant is your peracetic acid. However, peracetic acid is effective if it is used with an organic material. So, it is commonly used for surface sterilization of surgical instruments. So, glutaraldehyde in the form of your formaldehyde and peracetic acid is referred to as cold sterilization because you do not need to employ high humidity or heat. So, we're done with sterilization. Let's now go to physical methods of disinfection. You have three more common physical methods for disinfection. One is boiling at 100 degrees Celsius for 15 minutes, and this kills vegetative bacteria. Another is by pasteurizing for 30 minutes at 63 degrees Celsius. And this temperature and contact time kills food pathogen. And then sometimes you can use non-ionizing radiation such as your UV light, which is also seen in laboratories and other biological safety cabinet. Okay, other physical methods of disinfection also include non-ionizing radiation. So, kanina, for sterilization, you use gamma. But in disinfection, you use only UV light. UV light has long wavelength and it has low energy. It has limited sterilizing power because of poor penetration in most material. So usually you use this in certain areas of the hospital such as your TB laboratories, your operating rooms, and organisms should have a direct surface exposure. So it does not work if the surface that you want to sterilize is not exposed to this kind of radiation. So, makikita natin ngayon, maraming bumibili ng UV light for killing all forms of microbial forms seen in contact surfaces or fomites. However, take note that these fomites, if not exposed directly to the UV light, is not killed by the process, or is not, the surface is not disinfected. Other chemical method of disinfection can be accomplished by the different classes of chemical disinfectants, including your alcohols, your aldehydes, your halogens, your heavy metals, your quaternary ammonium compounds, or what we usually call your quats, and your phenolics. Okay, so using the chemical methods of disinfection, you have some um, uh, things that we have to define in terms of chemical disinfection. When we say chemical sterilants or biocides, you use the chemical to destroy all life forms. Biocides or chemical sterilants are usually used for shorter periods. And they function, if they are used for shorter periods, they function as disinfectants. Okay? It does not kill the spores if used for shorter periods. However, if you say antiseptics, these are disinfectants that are used on living tissue or what we usually apply on skin. What are types of antiseptics? So usually you use 70% alcohol and the like. So there are factors that influence the activity of disinfectants. Of course, the types of organism that you want to eradicate, the temperature and the pH during the process, the number of organisms present. For example, there is a lot of microbial load, so you would need more contact time if you're going to use a chemical sterilant. Okay, the concentration of the disinfectant where you will deem it effective, the amount of organic. For example, some sterilants or disinfectants may not work if there is a lot of blood, mucus, or pus present in the item that you want to disinfect, the nature of surface to be disinfected, if it's porous or non-porous, the length of contact time, and last but not the least is the type of water available, if it's hard or soft. This slide will show you the resistance of disinfectants vary with the type of microorganism present. You will note that the most susceptible to disinfectants would be your enveloped by viruses, such as your hepatitis viruses, and the least and most resistant organism are the bacterial spores, which is your Bacillus species, old with endospores. That's why they cannot be easily eradicated and they may pose as contaminants in all your microbial studies. Okay, so ngayong pandemic, palagi tayo may dalang alcohol. So, ano nga ba ang effective concentration ng alcohol? So, both ethanol or isopropyl or isopropanol are widely used. And normally, they are prepared at a concentration of about 70%. But both are non-sporicidal. So, ibig sabihin, they cannot eradicate spores. Their concentration of 60% and above up to 90% are effective. Okay? So, they are bactericidal and fungicidal but not effective against your endospores, non-sporicidal. Alcohols enhance the effectiveness of other chemical agents if used as disinfectant. Another commonly used disinfectant is the halogens. And a good example of halogen is the iodine and your chlorine. They are commonly used in the laboratory and in the household. So usually they are prepared as a sodium hypochlorite solution or household bleach to clean tabletops and blood spills. They are also recommended in COVID laboratories for disinfecting tabletops and biological safety cabinets. Okay, usually chlorine or iodine is followed by follows an initial disinfection with 70% ethyl alcohol. It is usually used when you are trying to do blood culture and it's used to disinfect the skin surface to deter growth of normal skin flora that is usually a problem for your blood culture samples. Okay, another sample is your phenols or carbolic acid. They are stable. Phenols injure plasma membrane. They inactivate enzymes. They denature proteins. And phenols like phenylphenol is a main ingredient in most formulations of your Lysol. So you usually commonly use this in the household or in the hospital setting. So we're done with disinfection. Let us now go to laboratory media. Okay. Laboratory media are the culture media used in microbiology. They are prepared either in semi-solid, solid, liquid formulations. And they aid us in facilitating growth of microorganism. So culture media in the microbiology lab provide sources of carbon, energy, nitrogen in the form of carbohydrates and amino acid for the growth of various microorganisms. Special media provides not only specific requirements but also include some inorganic salts or particular growth factors which are used to grow more fastidious organisms in the laboratory. So we have different types of media. The general, enriched, selective, enrichment, differential, transport, and we will differentiate them one by one. Okay, general media usually supports the growth of most organisms that do not have special nutritional requirements. Examples of general media include your nutrient broth, peptone water, and nutrient agar. they will grow almost anything that do not need special nutritional requirements. So enriched media naman has additional substances such as addition of blood, of egg, of serum. They are usually used for cultivation of fastidious organisms that cannot grow on special media alone. The use of culturing, they are usually used in culturing sterile body fluids to enhance the growth of organism, and are usually used for primary identification of some organisms such as your streptococcus. By using blood agar plate, you can see hemolysis patterns. Examples of enriched media, as we have mentioned, is your blood agar plate, your chocolate agar plate, and the Loeffler serum. The blood agar plate usually exhibits hemolysis of your Strep pyogenes. Okay, your chocolate agar plate supports the growth of your Neisseria meningitidis, your Haemophilus, and your Streptococcus pneumoniae. And the Loeffler serum, which is a semi-solid media, is usually used for growth of Corynebacterium diphtheriae. These are samples of your enriched media from blood agar plate showing hemolysis to chocolate agar plate in the middle, and your Loeffler's medium or serum used to favor the growth of Corynebacterium diphtheriae. When we say selective media, selective by the name implies they have usually they are usually in the form of solid media, and they contain inhibitory substances such as your bile salts, chemical dyes, antibiotics. They inhibit the growth of one organism and allow the growth and favor the growth of another. Selective media is usually used for culturing a specimen from a site having predominant normal microbial flora to prevent unwanted contaminants. For example, a media containing antibiotics that can deter the growth of fungal colonies and favor the growth of bacterial colonies is a selective media. The following are samples of selective media, such as your Leuenstein-Jensen medium, which is used for the growth of mycobacteria. It has an inhibitory malachite green. You have the modified Thayer-Martin that has vancomycin and nystatin and colistin. Usually, it inhibits most fungal organism and fastidious or normal flora, and they are usually used for the culture of Neisseria species. The TCBS is usually used to grow your Vibrio species. The inhibitory substance here is your bile salt and thiosulfate, and it inhibits the growth of other gram-negative organism and favors the growth of your Vibrio cholera. The XLD agar usually contains lactose and the desoxycholate, and is usually used to favor the growth of Salmonella and Shigella species. The SSA, as the name implies, also favors the growth of Salmonella and Shigella and inhibits other enterics. When we say enrichment media naman, usually this is a fluid media. It contains substances which favor the growth of organisms of interest. Usually, enrichment media is a preliminary step before you subculture into solid media. Ano ba yung mga examples ng enrichment media? One is your selenite broth, which you use for primary culturing of specimen, it will allow and favor isolation of Salmonella and Shigella, so that when you subculture, you can isolate them better. You can also use tetrathionate broth. Also, you can use that as step media for growing or isolation of Salmonella from fecal specimen. And then also, you can use alkaline peptone water to favor or enrich the growth of Vibrio species. Okay, differential media. The name itself, differential, they allow phenotypic differentiation by exhibiting certain culture characteristics of organism in plated media. They usually contain dye, indicator substances to morphologically differentiate the organism. The indicators change color when acid is produced following fermentation of a particular carbohydrate, and this is true for your Enterobacteriaceae family. So a good example of a differential media is your McConkey agar, which has a neutral red indicator, and it will differentiate the lactose fermenters appearing as pink colonies from the non-lactose fermenter, which will appear as colorless organism in the solid media. When we say transport media naman, usually transport media is a semi-solid media, and it has ingredients that prevent the overgrowth of commensals and ensure the uh, survival of aerobic and also anaerobic organisms. Examples of these include your Carrie Blair medium, your Amy's transport medium, and your thioglycolate broth for anaerobic organisms. Last but not the least is anaerobic culture media. Anaerobic needs a strict oxygen-free environment. So you have to culture anaerobic to be able to isolate anaerobic organism. You need an oxygen-free media. So anaerobic culture media are richer in organic compounds. They contain reducing agents such as cysteine and hemine. They have a redox indicator, and usually they are equipped with an anaerobic chamber and a gas pack to remove excess oxygen within the area of the media. So I do hope that you learned a lot. We have a lot to cover, and we have suggested some um, reading materials for you. So you can also browse some materials and your ebooks for reference. So thank you very much, and this will be followed by a laboratory tutorial and question and answer portion in your general lecture room. Thank you very much and good day.